This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 25 for March 20th, 2009. From New York City, I'm Vincent Racaniello.
And from Massachusetts, I'm Alan Dove.
And joining us today from sunny California is Luis Villarreal. Welcome to TWiV, Luis.
Oh, very happy to be here, although it's not sunny today.
I was just going to say, is it in fact sunny? I'm sorry. Is it?
It was yesterday, but we have an overcast. It does rain on occasion.
Irvine, California.
Yes.
University of California, and you're a professor in molecular biology and biochemistry. Is that right?
Yeah, and I direct the Center for Virus Research as well.
And I've known you for many years, right?
Absolutely.
But haven't seen you in a long time, so it's good to get you on this conversation, which we've been doing since September or so. We have a lot of fun.
Happy to join.
Trying to get all my virology buddies involved. So today, as usual, Luis is joining us to talk about viral evolution, which is something you like, right, Luis?
Yeah, it's a topic I've sort of had as a hobby, as an intense hobby for some decades now. So something I've been invested in for a while.
You have a nice book called
Viruses and the Evolution of Life.
Right.
Although there's a new book that just came out this year on having to do with origin of group identity, how genetic parasites have affected sort of group mechanisms in biological systems.
Is that also an ASM book?
No, that is not an ASM book. That's Springer. There's a book that I recommend for people who are interested in this topic is The Origin and Evolution of Viruses, which is a compiled book put together by Esteban Domingo, Colin Parrish, and John Holland. It came out this year, Academic Press. And I think that's the most sort of complete compendium on the topic, in my judgment.
Oh, okay. Well, we'll put a link to that in the show notes, and maybe we'll bring it up again later. So the ASM book, you're not planning to revise?
I haven't thought to do it. It probably could benefit from it, but that's not a priority project right now, but maybe.
Yeah, it's a lot of work.
Yes.
Well, you also had a nice Scientific American article 5 years ago, I guess now.
Yeah, that actually continues to get attention, and it's curious because that the assertion of that article is basically in opposition to this review that you mentioned, and it's curious how things. We'll talk about it, I guess, later, but how certain patterns of reference are sort of ignored.
So that's why Luis is here today. And but before we get to evolution, there are a couple of interesting stories in the news, and I think Alan, you should start with. I shouldn't laugh because these two people died in one of these stories, but
yes,
the first one, furious rabies after an atypical exposure.
Furious rabies, yes. It's not often you get to talk about a disease like furious rabies, but these classical designations. There are three types of rabies that you can present with. It's all the same virus, but it can present in three different ways, and the sort of classical presentation of it is called furious rabies because people tend to get agitated, and it also is accompanied by this very diagnostic feature called hydrophobia, which you've probably heard of. So furious rabies, and then there's also what's called dumb rabies, which is a paralytic disease. Again, same virus.
That's what I have, I guess.
Yeah, I think I've been suffering that for years. And then there's atypical rabies, which I guess is, if you don't present with one of those two, but you still have rabies, then it's atypical. So this is furious rabies. There's a case report in PLOS Medicine, that's Public Library of Science Medicine, an open access journal. And this just came out this week. And the atypical exposure, rabies, you normally get it from a dog bite or a raccoon bite or a bat bite or something like that. These are two cases from Viet Nam where the patients, in both cases, unrelated people, they just showed up at the hospital at various times and they had rabies, but they didn't have any history of exposure of a dog bite or any sort of animal bite. But it came out in getting the patient histories that both of them had eaten delicacies of Vietnamese food. In one case, the patient had butchered and eaten a dog. And in the other case, the patient had butchered and eaten a cat. And it appears that in the butchering process, they got rabies. There were other people. So in first patient had taken this dog that had been hit in a road accident. So it was roadkill. And it's difficult. I mean, both of these patients died, so we should keep this in perspective. These are tragic cases. But in Vietnam, it is traditional to eat dog or cat. And in this country, a lot of people make fun of this, but if you think about some of the things that we eat, I'm from Maryland, where it's perfectly normal to eat oysters and crabs, and a crab is a giant seagoing bug, so how weird is that? So in Vietnam, people will eat dogs, and he had this dog that had been hit by a car, so he took it home and butchered it, ate it, and also other people ate it as well. He cooked it for a group. He was the only one who got rabies. So apparently something in the butchering process carried the disease to him. And then the other patient, similar story, had a sick cat, killed it, butchered it, and then ate it along with several other people. None of the other people who ate it got the disease, but the fellow who'd butchered it got the disease. So it appears that butchering animals is a risk factor for rabies. There was some other work on this, and it popped up on my radar. Here's a type of exposure you don't hear a lot about, and hopefully you won't encounter it very much.
Both of them probably got it by aerosol, I would think, because they both cut the skulls open and took out the brain.
Yeah. So I was going to get to the...
you're going to describe...
The butchering. Yeah, the butchering was not just that they skinned the animal and, took out the meat. This is apparently in preparing these dishes, they singed the hide to remove the hair rather than skinning the animal, then opened the skull to take out the brain. And I think with the dog, the brain was steamed, wrapped in leaves and steamed. And then with the cat, the brain was taken out and pulped. The patient took the brain out and pulped it in his bare hands and then cooked it up. So somewhere in the process of singeing the hair off the animal and breaking open the skull and pulping the brain with the hands or, wrapping it up, that's probably where the virus got out. And either through a small cut in the hand or through an aerosol, kind of a gruesome process. But
something along these lines was described in Darwin's accounts when he visited Venezuela and butchering cattle there, and some people were exposed to a rabid cow, and there was an outbreak in the camp.
Really?
Of rabies. Okay. So this kind of thing is actually, I don't know if you know, but I started my graduate education actually working with rabies as well as a few other negative stranded viruses.
Yep.
So at that time, I was very fascinated in many of these aspects, still am, and many of them are still mysterious. All kinds of issues sort of come into thought when you start thinking about this virus and its relationships with its host and the consequences. For example, what does it mean to be a carnivore and eat raw meat all the time and expose yourself to all these agents? It seemed like it would present that life cycle with a particular sort of risk factor that, something that only eats plants probably never encounters.
Yeah, no, I mean, in places where the virus is still endemic in various species, that's an issue, right?
And this capacity to induce anger and rage, and it's also associated with hyper-awareness during this period of illness, to me has always been a fascinating CNS phenomenon that's really poorly understood. I would like to know more about it, but there's not much in the literature that deals with it.
No, that's a good point. Nobody really understands that. Two interesting things, I guess. First guy and his wife, they both said he had not been bitten by an animal. So they didn't even connect eating this dog with getting rabies, which is interesting. That's a matter of education, I guess. And then both of them, patient 1 took 2 months to incubate, and patient 2, 3 weeks, which I think is consistent with a peripheral inoculation, taking a while for it to reach the CNS, right, Luis?
Yes, that's the usual view. But, aerosol exposure of laboratory workers was confirmed years ago. That's happened. And also aerosol exposure in bat caves is known to have happened. So people who work with the virus and have to deal with large numbers of it have to be concerned with that potential avenue of exposure.
So if you inhale it by aerosol, what kind of incubation time is that?
Well, most of these cases are very rare and uncommon, and trying to reconstruct the progression of the disease is difficult. But it's on the scale of what you mentioned, usually 3 weeks to 2 months. Yeah. You tend to forget what has happened during that period, or You have to sort of reconstruct after the fact.
Sure.
I'm also fascinated very much, I don't know if you're going to talk about it, by the occurrence of occasional survivors of rabies, which used to be thought never happened. And in the last few years, there's been at least a few cases that...
Yeah, we actually brought it up a couple of weeks ago. There was, as far as we could tell, there's only been one case in an unvaccinated individual, and it was a person here in the U.S. who got rabies, and she was put into a coma by her physicians and ended up surviving, and somehow it's thought that being placed in a coma had something to do with her survival. And now she's apparently fine. She was debilitated for a long time, but went to college even. And all the other cases were people who had been immunized, but apparently the vaccine didn't take well, and so they got sort of a partial disease and didn't die.
But that just relates back. Somehow this virus has some particular capacity with respect to what it's doing to the CNS in terms of, this coma, which I guess is questioned, but there's no way to repeat that experiment.
Yeah, sure. We don't know if that had anything to do with her survival, right?
Yeah, but it's such a rare event that you can't dismiss it as a possibility. Anytime you have such two very unusual occurrences, you can't just assume that they're linked.
We should point out that in these two cases, they came into the hospital with symptoms. In that case, it's already too late to immunize, right, Luis?
Yes, typically, yes.
Right.
That's
Right. Whereas if you got them right after they'd been exposed, then it might have been a different story.
Yeah.
Yeah. So rabies doesn't go away, and the fact that it doesn't go away is really a topic that I'm ultimately the most interested in, because my long-term interest has always been persistence.
Mm-hmm.
And this is the theme of my book, how I think about the long-term evolutionary biology of viruses. And in the context of rabies, for example, we know it has persistent relationships with specific hosts, specific bat species, for example.
Yeah.
And from my perspective, we know so little about that relationship. We just put a label on it. We say, well, it's a reservoir, as if that helps us understand anything, and just dismiss it, where it has a completely distinct relationship with its host, in that it's there typically for the life of the animal without any disease. Clear potential for transmission, but we don't know anything about the selective parameters or the molecular functions of any of the proteins in the context of this tissue.
Is it neurotrophic in bats?
Even that isn't clear.
Okay.
The assertion has been made, but I've never seen compelling data on that. It's very difficult to actually localize this sort of a latent or persistent infection. This usually takes a significant investment of resources.
But it's probably hard to do in bats anyway, right?
Yes. So, I used to conduct these international programs where we go down to like the rainforest in southern Mexico and involved with mammalian biologists that would collect bats and so forth. I was amazed at how little concern they had with the kinds of viruses that they were handling. Since I was taking students down to these things, I thought, well, maybe we don't want our students handling these bats with bare hands.
We've talked about bats a lot on TWiV because they harbor, as you know, other viruses, Nipah, Hendra, and probably the Ebola viruses as well. SARS. So you're absolutely right that we really do.
In all those cases, though, if you say, what does the glycoprotein X in Ebola or SARS, why does it have a particular transmembrane immunosuppressive domain that's associated with that species? Because this is what ends up being a problem when it jumps species like into humans and is capable of causing some pretty much unpredictable start pathology. The point being is we don't really understand the evolutionary forces that are maintaining it in this state. We just assume that they're the same, and in my judgment, they're clearly not. The big unknown.
Yeah, that's a good point. There was one other thing. Oh, yes. So I looked these cases up on ProMed Mail, and so they say that these animals weren't available, and so we don't actually know if they were the source of rabies because no one was able to look for it.
No,
because the animals had been eaten.
Yes, that's right.
The experiment was eaten.
Yeah, so, take it with a little grain of salt, but I guess since there's no other indication of how they got the virus that you would have to.
And frankly, if I were eating dog brain, I'd take it with a large grain of salt.
Yes.
Yes.
So the moral is don't eat your pets, right?
Right.
Don't eat your pets or your neighbors.
Now, when you posted this the other day on Twitter, Alan, I just happened also to notice there was an article in the Times about a rabies outbreak in Angola.
That's right.
Rabies outbreak in Angola caused by roaming dogs kills 93 children. Angola, of course, being in the southern part of Africa. And, in a lot of parts of the world, there are still dogs running around with rabies. Besides the bats, there's still plenty of infected dogs. In a lot of countries, you immunize your pets against rabies, but there are wild dogs running around. I guess you have to have a program to immunize them. Yeah. When money is short for a lot of things, the dogs are not the priority.
It's such a horrible disease too, so it's really a tragic occurrence. Yeah. The resources for immunizing a population of dogs like that are really rather minimal. But
yeah. I mean, in this case, in fact, they said that the kids could have been saved because a lot of them
knew about the exposure.
Yeah, and they didn't have vaccine. Apparently, there's a shortage of vaccine. Sanofi Pasteur, who is the supplier, they have a factory under renovation, and so they couldn't get any in time, which is really bad. Kids dying of rabies is bad.
It's horrific.
But they said one thing in ProMed Mail, which is interesting, that a local person in Angola had said, we're catching all these dogs and we're looking for rabies. And ProMed Mail says, although there is a test for rabies in humans, the equivalent test is not available in dogs. You have to take out CNS tissue, basically. So the only thing you can do with the dogs is to immunize them, not to look for rabies in them. Just immunize all of them. I guess you could. In some countries, they put vaccine-laden bait out, right, Louis?
Yeah, that seems to be pretty effective. That's done in the United States as well for wild populations. So that is effective.
So Bill Gates should donate money for this.
I think so. I think this kind of phenomenon should not be happening in this century.
Nope.
Well, and if you're using baits, you don't have to catch the dogs. You don't even have to know where they are. You just go through a neighborhood and throw these things into corners, and the dogs will eat them.
Yep.
By the way, the baits, this gets into a topic that we might want to talk about. The baits use live virus, right? This concept of live versus dead virus or inclusion in the tree of life and so forth is one that we might want to get into.
Yeah, we shall. You bet we shall. Then there were a couple of stories this week, which I thought were interesting, about scientists sticking themselves with needles that have viruses in them.
Ooh.
And there was one in which case there was a scientist, I believe, in Germany who stuck herself with a needle with ebolavirus in it. Right. So this was reported in Science and also ProMed Mail. It happened, I guess last week. Right. So I guess she was inoculating mice, and she stuck herself. And, there were probably at least 1 million virus particles in there.
I assume this was in a BSL-4 facility?
Correct. Institute of Tropical Medicine in Hamburg.
Yeah, handling a sharp in that facility. That sounds like the worst possible circumstance.
I mean, that's the thing. If you want to put virus into animals, you have to use a syringe. But an accident will always happen, right? People are fallible, and there's nothing you can do until you make a latex glove that is impermeable.
Yeah.
I suggested that we get the Cylons to do this, but because they won't make mistakes, but that's not an option here. So then this is interesting. So here you have people working on the virus. Your colleague gets stuck. What do? There aren't any antivirals. So they decided to give her an experimental vaccine, which I think is a VSV pseudotype with the Ebola glycoprotein.
Is that it? I thought one of the ones I had heard of is the recombinant adeno one.
That is a DNA-based one? Is that?
Yeah. I don't know the status of it, though.
They mention it here. They said they considered it, but do you remember this, Alan, why they decided to use the pseudotype?
Yeah, they didn't.
Had they have results in primates with any of these?
Well, the pseudotype, was
right. The VSV they had results from primates, and I guess that was the basis.
They convinced them, yeah.
So the DNA vaccine was never tested in humans. I don't think the VSV one was either.
No, it wasn't. Neither one's been tested in humans.
Why not use them both? They don't interfere with one another.
Yeah. So they gave her the VSV pseudotype, and she developed a fever the next day, which could have been the vaccine or Ebola. And now the fever is gone, and she's still in isolation. Now they're debating whether they can bring her out or not. They say she's depressed being isolated.
How long has that been?
Since last week, I guess. So it's just a week. So maybe she'll be okay because, the Ebola infection didn't necessarily have to go anywhere. I don't know what strain this was. They don't actually mention it in the ProMed Mail. note. So, if it were Reston, then it should be okay because it's not terribly virulent. But
big difference.
So here's the question. If it were you guys and you stuck yourself, what would you do? Would you take a vaccine or?
Yes, definitely. Would take all the experimental vaccines. And if there are any antivirals, throw them in as well. And just hope to wait it out. That's
I'd throw everything at it and just assume that I'd be isolated in the unit for a while.
I don't think she should worry about being isolated. She's happy to be alive, probably, right?
Yeah, exactly.
Well, everyone reacts differently, so we shouldn't be critical of that. But it's going to happen again. And in fact, there was another incident, this time a West Nile laboratory-acquired infection, which happened some time ago, and it was just published on March 19th in New England Journal of Medicine. This was a South African scientist stuck herself with a needle with a neuroinvasive West Nile virus strain, and she put in 50 TCID50s. She got sick 7 days after. She got backaches, neck stiffness, malaise, etc. So they decided to look at her cytokine response. Because, it's probably, here she was 29 years old, immunocompetent, so she probably isn't going to get very sick from this. So they decided to look at her cytokine. So they made an experiment. And in fact, this paper is reporting her cytokine expression. They looked at a bunch of different ones at different times after infection.
Did she at least get to be an author on the paper?
I don't know they don't say what her name is. They have 5 authors. All from South Africa, a couple of female, it looks like, maybe all of them. So I don't know if one of those is actually the patient because they just call her a 29-year-old female scientist. So maybe, I hope so.
Yeah.
Was there anything unusual about the cytokine profile?
It had an unusual increase in interferon alpha, which they say, maybe that's why most people control it well. And this could say, well, maybe we should use interferon to treat these infections in compromised people. They conclude by saying the cytokines may serve as additional targets for development of therapeutic interventions by suppressing pro-inflammatory responses or supplementing type 1 helper T responses. Probably one of the few cases where you have a course of cytokine induction in a human infection. The time zero, right?
Right.
Yeah.
If you stick yourself, you might consider doing an experiment.
Well,
there was a time not long ago when it was the other way around. You'd consider doing an experiment so you would stick yourself.
Yeah, that's right.
Which I did with the smallpox vaccination when we had this anthrax attack, and it was apparent that we sort of caught with our pants down with respect to our smallpox vaccine.
Oh, yeah, you inoculated yourself?
I had myself inoculated, and I collected blood so that we could get a whole set of data out of that. And it was very useful information, actually.
Did you publish it?
Yeah, it is published. It's been published. If you look it up under my name, you'll see that there was this series of human studies with vaccinia and looking at the immune profile that results from that. That was me, basically. At least that was the first one.
Well, many scientists, I think, do this. I knew a Russian scientist, the guy who discovered hepatitis E virus, and he actually drank stool filtrates to prove that they contained the virus and gave himself hepatitis. It's not a very nice picture, right?
No, that's a very committed soul.
Yeah, my grandfather did that with creeping eruption. Proved that the etiologic agent was cat hookworm. Gave himself a case of it.
Well, I think as we work with more dangerous viruses, this will become less likely. But many people now don't even work with live viruses. It's all genomics and proteomics.
And I think that never gives you the information you need with respect to disease and the intervention in people. And for example, I'm getting very interested in hemorrhagic fever with dengue, and that's a specific phenomenon of humans. And you might build a model to try to emulate it, but you never know how accurate that model is going to be.
Sure, absolutely.
Right.
Yep. Now, an N of 1 is typically also not a good experimental design.
No, but it could be a very big clue.
It could.
Yes.
Before we go on to our main topic for today, let's have a brief message from the American Society for Microbiology.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th General Meeting, the largest annual gathering of microbiologists in the world. Visit the General Meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
Okay, so that is our story set for today. So let's move to evolution. Let's begin evolving, Luis.
Oh, great.
Where do we start?
I think we can start just by thinking about how viruses affect life on this planet and whether viruses have features of life. Whether you define them as living or not is operationally, in my judgment, irrelevant, which was mainly the theme of the Scientific American article I wrote.
Mm-hmm.
And in picornaviruses, this is a particularly sort of fruitful system to think about. You're probably familiar with some of the things that Eckhard Wimmer has written along these lines, in which he's called poliovirus a living chemical in a sense, because trained as a chemist in Germany, he looks at things from the perspective of a chemist. But so he'll put up this formula weight with all these numbers for carbons and hydrogens and nitrogens and so forth. But of course, we know that formula weight is fake because anytime you propagate a virus like poliovirus, because of the error-prone replication, you always generate a diversity, a quasispecies. We can talk about that later. And it is that capacity, that error-prone capacity of replication, which allows something to transition from this inert chemical state to a state of something that takes on features of a living system. So we know that with polio, you can take what's called a quasi-dead template, a template that by itself is not capable of propagation. And if you allow it error-prone replication, one of the progeny will be capable of setting up an infection and being transmitted, although the original template doesn't have the capacity. So in a sense, this process of error-prone replication can endow a chemical entity with some of the elements of a living system. And that's sort of a basic assertion that I made in my first book, Viruses in the Evolution of Life, where sort of thinking about early life must have been these prebiotic, precellular replicators. Anything that replicates, anything that has the capacity to sort of specify its catalytic copying, as well as the information to specify its information content, is going to be prone to parasites of that process, and that's basically what a virus is. I think it's a really a foundation issue in terms of when you start thinking about, okay, what does it need to get going with respect to life? And as soon as you start thinking about that, you see that viruses are sort of involved. We can sort of extend it from there in terms of how we think this applies to our current thinking.
Could I ask you one question? So you talked about seeds in your Scientific American article.
Yeah.
So we were discussing this a couple of weeks ago. So my point is viruses are not living because they don't do anything till you get in a cell. And then I think Alan said, but seeds and spores are the same. So how do they fall?
That's equivalent point, and that is if you have a chemical entity like a virion, just like one that Eckhard Wimmer might have made in a test tube, right, with kind of a complicated mixture of stuff. It's as dead as a doornail. It's a chemical entity. It has none of the features that we would consider to be fundamental to life until you put into a more dynamic system. But that dynamic system can be an in vitro system, as Eckhard Wimmer showed. So it doesn't necessarily have to. It has to be more complicated and provide energy and a few other things. But when you get to if you apply these same concepts to a viroid, that's catalytic RNA, the requirements are significantly reduced, but you still end up with a system that starts to acquire some of the features of life as a product of error-borne replication, and it becomes capable of selection and adaptation. And then it takes on these features. So it's true, a seed and a virion are more or less in the same state, a state of just sitting there not doing anything. And we know there's lots of biological systems that have the capacity to sit like in an ice shelf for maybe 10,000 years and not do anything, but they've maintained the capacity for life. And in a sense, I think this is what persistence gives to the maintenance of a viral lineage. It's the capacity for continuation. It's the capacity for life. Not necessarily that it has made a lot of progeny. It may have made no progeny or one progeny, but it's the maintenance of the capacity, which is what's being selected for. So in the context, for example, of these bats that are making very little virus but do spit it out on occasion in unknown circumstances, somehow there's been this selection and evolution for the maintenance of this capacity. And I think this has had big consequences to what lineage lives and what lineage dies. I think this has big effects in terms of populations that prevail and populations that succumb.
You think that the viruses may help the bats in some way?
Yes, and let me just explain that a little bit, because this idea, I think, is counterintuitive and many people balk at it. And since we work with mice a lot, they're a very familiar topic and we're comfortable to think about them. let's say I go outside and I catch a wild Mus musculus domesticus, and I bring it in here to my laboratory, and I want to take it downstairs and take it to our mouse colony. Well, anyone who's ever tried to do that will encounter basically a fit by the veterinary staff to say, that thing's contaminated with 20 different persistent viruses. You bring that in here, you're going to destroy our colony. And that's correct. We know we can pick one specific example, like mouse hepatitis virus, which is a coronavirus. And in field studies, it's basically 100% prevalent in these wild animals. And so it's reliable. You bring them into a colony, you're going to wipe out your colony. Yet, that persistently infected population of wild mouse is going to be fit, it's going to be healthy, and it's not going to have no problem for the most part. You can create problems, but mostly it's going to be healthy with all these different viruses. Mm-hmm. So you can see, based on this model of population interaction, that when two populations come together with different composition of viruses, those viruses that are sort of incompatible with persistence in the new population can really do a number on them. Right. And we see these kinds of phenomenon occurring. That's basically what emergence is, right? Emergence of a new disease from some persistent state to wipe out or affect in a strong way a population of a different host. Sometimes it's just the same species, sometimes it's a different species, but it does affect how populations compete. Mm-hmm. And I guess that would be my main assertion, that this has big consequences. Whether you're growing E. coli, whether you're growing lactobacillus, whether you're growing fish, plants, you name it, anybody who grows life in a large sort of manner has to be very concerned with the sources of persistent viruses they're bringing into their population, because really bad things tend to happen. And this is just an operational fact that we live with. But the evolutionary implications of that are kind of lost, because we dismiss viruses basically as having any role in terms of host survival, other than just being consumers of host.
Well, of course, in the ocean, at least the marine virologists know or understand that they have a big role in the ecology and geothermal cycles, for example, right?
And that seems to be true in the soil as well, and in hydrothermal vents, and pretty much any habitat that's intensely colonized with life, you see evidence of a rather big role for viruses.
Are there viruses at the hydrothermal vents?
Yes, absolutely. And their counts are surprisingly high, and they have a composition and a nature that's completely distinct. You might want to talk to somebody who works in that area sometime. That would be a fascinating show.
Definitely. Because last week we were saying, are there viruses at the hot vents? And we didn't know.
Yes,
so, great.
In huge quantities. But not only that, but they're a completely distinct set. And they're shapes and kinds of viruses that look bizarre by most assessments. Wow. You see things and shapes that you never see, and relationships that are quite distinct. They're like with the hypothermophiles, they're predominantly chronic persistent infections rather than these lytic acute infections that you see with sort of TWiV and phages and the alpha proteobacteria. Mm-hmm. So it's really a distinct relationship in these archaebacteria and their viruses than what we tend to see in the prokaryotes.
So let me just make it clear. What you're saying is the kinds of relationships that the marine virologists detect in marine viruses in controlling marine populations, microbial communities, energy recycling. You think that happens in all biomes, basically?
Yes, but the relationships are distinct.
Sure.
And they're specific to the biome. And this is a curiosity. And, the relationship between a virus and its host is like a fractal. Whether you look at it at a very sort of broad scale or a very detailed scale, you still see that there's this peculiar and specific relationship. For example, why are there no RNA viruses in archaea that we've been able to describe so far? Here's this whole domain of life. Mm-hmm. The coronaviruses, for example. That would seem like a very fundamental viral strategy that should have been around since before the beginning. Yet we see in other domains of life, like in flowering plants, it's positive stranded RNA viruses are as ubiquitous as can be. There are so many types, and they're interacting in all these complicated ways. So here we have these two distinct biomes that have the very different fundamental relationships with the kinds of viruses and my assertion is that's always the case. If you're looking at ferns, if you're looking at algae, if you're looking at bacteria, if you're looking at people, they all have their own peculiar relationships with the kind of viruses that they support, particularly the ones that are capable of colonizing them in a persistent way. So that's one of the assertions I make in my book, and it's an assertion that I sort of continue in my new book, The Origin of Group Identity.
So where in the tree of life would you say we should put viruses?
There is no part of the tree of life that doesn't have significant virus influence. And I was thinking about, I know this person who wrote this review because he was in the audience at a presentation I did in France about 5 years ago. And so it's curious to me that it doesn't reference a whole body of literature that basically counteracts his assertions. And what I suspect is the case is people who come from certain perspectives or backgrounds, particularly the evolutionary biologists, really have set views of what they think viruses are. And I find the virologists are more open to interpreting the meaning, significance, and interactions between viruses and their hosts. Because we realize there's all these relationships that we poorly understand, and there's interactions that are kind of complicated, and we don't pretend to understand them, but it affects our basic definition of what we think a virus is.
So would you map the viruses parallel to the organisms that they infect, or would they be a separate tree superimposed on?
Well, you clearly can't separate them. If you look at our genome, if you look at the genome of E. coli, if you look at any comparative genomic analysis, the most dynamic part of the genome of any living entity that I'm aware of is manipulated by viral action.
Right.
So the difference between us and chimpanzees is mostly to be found in the HERV patterns and LTRs and other parasites of parasites that really colonize the two lineages. The difference, if you go to the ECO collection of what used to be 70, I don't know how many it's up to, perhaps 100 now, sequenced medically important genomes, they really differ by prophage colonization and blocks of genes associated with tRNA insertion points. Clearly phage-mediated, in my judgment, to a large degree. So it doesn't matter what end of the spectrum, what organism you're talking about. If you take a strain of mouse, say it's a BALB/C or an AJ or whatever you choose to look at, you can characterize it based on its pattern of, say, endogenous retroviruses that it has or expresses. It's pathognomonic for the strain. That's true of sheep, too. So my point is, it's silly to have an argument that viruses are not a component of the tree of life when you see their footprints so abundantly in all genomes. And for this thing to have been published, I think, in Nature, just is disappointing scholarship. At least it should have been rebutted by somebody. I don't want to rebut it because I'm lazy, but somebody else may want to do it.
Well, I think we should mention that what we're talking about here is a paper in Nature Reviews Microbiology, which we'll put a citation in the show notes. It's called 10 Reasons to Exclude Viruses from the Tree of Life. But you should rebut it, Luis. Well, you're doing it here, so we'll send people to TWiV to rebut it.
Well, yeah, not only that, but he was aware of this book I wrote, which presents many of the rebuttals, and didn't refer to it. So that's just bad scholarship.
So one of the things he says is that you can't put viruses ancestral to life. What do you think about that?
Well, it is problematic to trace the ancient history of something like a virus because they have these mosaic and quasi-species nature. They don't have a single lineage. Mm-hmm. Yeah. And you keep fooling yourself. We make these dendrograms that we publish in the literature all the time where we assume we understand a particular lineage of a virus as we trace one particular thing, say a polymerase or a capsid protein or an LTR. It doesn't matter. The thing is as you do those dendrograms with different genes, you always get variations of these trees. Yeah. And that's been true of the host for a long time as well. That suggests, more than suggests, it's only consistent with genes moving around in patterns different than that of their host. The only way you can accomplish that is to have an infectious agent capable of bringing genes in from external sources. We call this horizontal gene transfer. And really the only effective agents that do that are some type of virus, defectives or recombinants or satellites, or some mixture of viruses and the parasites of viruses. These LTR and various combinations that we know are prevalent.
So one of our readers actually asked a question with respect to that, because one of the points of this article is that viruses are polyphyletic for the reason you just discussed.
Yes.
And so I guess the thing to do would be to observe a new lineage emerging today. So he asked, could we observe a contemporary protovirus as it emerges?
Sure. It's happening right now. In fact, it happens all the time in real time. And how it happens is precisely this way. It's by the merger of other lineages of viruses for the most part. Viruses tend to evolve from viruses. There's very few, if any, examples I know of where you can point to the emergence of an agent that really comes from a non-viral source. In fact, I don't know of any examples that I think are good. For that assertion, even though that's a common view. It's a common view that viruses are these thieves of genes, and they just steal them out of the host, and then they put them to their use. Clearly, that can occur because there's no constraints on what viruses can use as genetic material. But it's also clear that when we sequence genomes of viruses, and You might want to talk to Forrest Rohr, who's doing metagenomic sequences of habitats and really has a big viral database that he's created. He's probably the number one person in the world on that. But it's clear that the genes that you're seeing in these lineages of viruses have no host counterparts for the most part. Mm-hmm. Like 80% of them. You can point to any large DNA virus sequence and make that assertion, and that's what you see. That whether it's mimivirus or fecodinoviruses or even baculoviruses or pox viruses, you see that there's a large set of genes that's usually peculiar to that lineage, from which you can find no host counterparts. That in itself, I think, is just evidence that, viruses can recombine at high rates, can mutate at high rates, can withstand lots of genetic turmoil, and they can explore sequence space at rates way beyond, 1 million-fold beyond what the host can. It's not a big leap of the imagination to say, gee, maybe they put genes together sometimes. And in fact, we know this has happened. But in the context of emergence, right now, I think the most fascinating emergent event is happening in Australia. And there's two of them that I'm paying attention to. One is with the koala bears, which are undergoing endogenization with a retrovirus that looks to me like it emerged from a rodent, although it looks like the gibbon ape leukemia virus as well. And the other one is happening with another marsupial species, the red-barred bandicoot, which is undergoing a near-extinction event associated with the emergence of what seems to be a recombinant DNA virus that looks like it's a part of a human papillomavirus and part of an avian polyomavirus. It's like got the capsid from one and the T antigen of the other. And these are having big consequences to these populations. We're talking about near extermination events. And if these populations survive, they're going to become endogenized and persistent and have a different relationship with this virus, which emerged from some exogenous source, is going to become part of their genome.
And people are studying this. They're sampling and watching it.
They are studying it, but the numbers of people involved and the resources involved are teeny relative to what we put into other projects. So I'm communicating with some of the folks who are doing this, and they're not too many. But to me, this is a huge event because these are the kinds of phenomenon that must have occurred recently in our human lineage. Although what happened in the human lineage is distinct from what we're seeing in the koala. It's a distinct phenomenon. Yeah, I think these are some of the deepest issues with respect to evolutionary biology. But you won't see the evolutionary biologists considering these events with the same kind of importance that I would.
So the people who are studying these events in Australia are virologists, then?
They're often veterinarians who have to take care of animals and zoos and stuff. It's the people who have to keep things alive, and suddenly everything starts dying, and you have to figure out what's going on. And that's typically how people discover these new emergent agents, and then they have to teach themselves something about how you figure out, what's important to learn as a virus that has emerged from these exogenous sources is now decimating a particular population. It's a species-specific phenomenon, right? This is not just wiping out all organisms. It's a particular species.
So viruses have so many unique genes, which is the observation that you just mentioned. Why don't we find those in cells?
I think you do, but typically the ones that entered cells are very ancient events, and therefore you don't see them as sort of current phenomena. That's mainly because I would argue that higher organism evolution is not gene-centric like it is in prokaryotes, where new genes occur. There, for example, if you get infected with toxigenic E. coli as opposed to laboratory E. coli, the consequences are hugely different. Right. But that toxigenic E. coli has been made capable of making you deathly ill because it has two phage that have colonized it and brought to it these toxic genes. And this is a common phenomenon in the prokaryotic world, that these agents bring in gene sets that sort of transform the selective capacity of the host. And in our case, it's a big deal, right? And so we know that this is more or less the way the prokaryotic world evolves and adapts. It does acquire genes in large numbers. Eukaryotic world, in my judgment, is much more regulatory in terms of how it adapts. So if you look at our brain in terms of the numbers of genes it takes or is expressed by some kind of transcriptional analysis, look at a chimpanzee brain or look at even a mouse brain, they're not all that different. But the regulatory complexity is vastly different with respect to what humans do and what these lower mammals do. And that's the product of what we would call all this junk DNA having acquired and manipulated the regulatory capacity of the system. And that's basically the action of or I would argue is stimulated by the action of these endogenous viruses, the HERV-K family and all these other families that are specific to human lineages, for example, and differentiate us from our chimpanzees in a very clear way, and are expressed in the neocortex during development at huge numbers. So we have all these footprints, and to assert that, okay, this has nothing to do with human evolution, like this article in Nature does, it's just silly. It's just ignoring a whole slew of facts and observations.
Is the emergence of HIV in the early 1900s, is this an example of protovirus, or is that just another zoonoses?
I would say it's also an example of a protovirus, but it fits more or less the same pattern, is that you have the fusion of two lineages, or two quasi-lineages of SIV that were adapted in something like a chimpanzee and then adapted to disease status in a human. It's curious that the primates are really prone to interactions with these exogenous retroviruses like the SIVs and the foamiviruses that are so sort of specific and prevalent to them. It's particularly the African primates, where other lineages you have a completely distinct relationship, like rodents, for example. You just don't see that kind of situation in natural populations, or marsupials. It's a completely distinct virus-host relationship. Something about the evolutionary history of the African primates in particular makes them really susceptible to persistent infection with SIVs and with foamiviruses. Foamiviruses, I think, are a really fascinating system, because here a virus that's lytic in culture, but virtually never lytic in its host, and its stability, its evolutionary stability, is astounding. It's equal to that of the cellular genes. Yet it's the same kind of replicator and system of error-based replication that HIV has. And so we have these millionfold differences in the rates at which diversity is being generated by a system that's basically identical. But really what's controlling this vastly different generation of a population or quasi-species is something to do with the biological relationship between the two, which is poorly understood.
We've brought up quasi-species twice, but probably a lot of listeners don't know what that is.
So, to me, the issue of quasi-species has recently taken on almost the equivalent of a paradigm shift. Let me define it as historically, in the 70s, Manfred Eigen, who was a physicist known for eigenvalues in physics, wanted to think about the circumstance for the origin of life based on the perspective of it being coming from RNA-based replicators. And he developed a series of concepts. One of them was this hypercycle idea, which we may talk about if there's time. And another one is this quasi-species idea, which is basically trying to describe mathematically the products of error-prone replication and how you can define it in terms of some type of selective coefficient of, the favored replicator, let's say. And so he made a series of assumptions about how you define this population and the characters it has, and express these in the context of a set of ordinary differential equations. These are the quasi-species equations, which basically define the heterogeneity and how it sort of the parameters of the heterogeneity of this population. And then that, coupled to Darwinian thinking, like selection of the most efficient replicator, allows you to implement it in the context of an evolutionary paradigm. A lot of people thought that, well, that's nothing more than a different expression of population genetics as done by Fisher, and that was an attitude that was held by many for some years. But experimentally, it interested a lot of basic RNA virologists, folks like Holland and Domingo and so forth, really started to do a whole new generation of experiments for the next 10 or 15 years, evaluating various aspects and parameters of this quasi-species concept, and ultimately came to a conclusion that some of the basic assumptions of quasi-species are clearly not realistic or correct. And a good summary of this is presented in this Origin and Evolution of Viruses. Esteban Domingo has chapter 4 in there, in which he talks about quasi-species, and he sort of makes these points very clearly. Basically, the experimental observations came to the conclusion that many of the foundations of quasi-species theory are not correct. There are interactions between the products. You can have complementation, interference, competition, and these have large consequences to the nature of the population. And there are other phenomena with respect to how the thing is developing that sort of violate the foundations. But nonetheless, this way of thinking created a way to to look at virus evolution that really is from population perspective, but a population in which selection may be occurring. In other words, population-based selection, which is supposed to not exist according to classical Darwinian thinking. And in my judgment, this really came to fruition experimentally when the Andino group did their experiments with the mouse model for poliovirus disease in terms of being able to manipulate the rate at which a polymerase has sort of fidelity, or whether you can throw in some analogs of nucleosides and manipulate the diversity of products that are formed, in which they come to the conclusion that it is the capacity to generate diversity per se that is essential for at least the pathogenic fitness that they observe in this animal model system.
So what you're thinking about is when he introduced a mutation in the viral polymerase that makes it less error prone.
Yes.
And then puts that virus into an animal. It doesn't do well because of its inability.
And that's using what's considered the master fittest average template, right?
Right.
Because the historic thinking of population genetics is that average represents the average most fit configuration. And when put sort of into this context, the readout is you need to generate variation in order to generate appropriate fitness. And that's really a different mindset than what the original quasi-species would have led us to. So it's a consortia. The outliers matter. If you have a defective or a variant or something that has a certain potential, it's going to affect the future of the population. And as we look at HIV evolution or hepatitis C virus evolution, we see that the outliers matter a lot. But we don't understand this very well because we don't have the very good tools for measuring the consequences of a quasi-species. If you just think about the diversity involved and trying to understand, well, what does it matter to have this particular version of the population versus that minority version of the population to the dynamics of the whole thing? These just present a whole slew of technical issues that we're not really prepared to deal with at this time. We just got to the point where we can perhaps apply pyrosequencing methods to get a snapshot of a quasi-species in a human infection with like hepatitis C virus or something like that. But in terms of understanding the significance of it, even if we measure in vitro that a defective interfering minority population has a consequence to something that you do in culture, what does that tell us about disease progression in a more complicated circumstance of a human? We are not in a position to evaluate a lot of these implications at this point, but I suspect this is what we're going to have to do to fully understand how quasi-species affects the survival of a population.
So is there a different role for quasi-species in an acute versus a persistent infection?
Yeah, there is good evidence that you can have sort of different dynamics with the same virus in the same host. And again, if you turn to mouse hepatitis virus, it's in the literature already that during a persistent enteric infection, you have rather narrow quasi-species composition, and during an acute infection, it diversifies considerably. It's sort of like the difference between the foamy virus in a monkey versus HIV in a human is that in one case, the quasi-species is tightly maintained, presumably by some type of selective parameters that we don't understand, and the other species is like fully open to explore sequence spaces as quickly as it can, which means you have this incredible hyper astronomical capacity for variation. So even the same virus in the same host, in particular circumstances, you can see, I think, pretty good experimental evidence that you can have these two different states.
In a person, let's say, has a common cold, is their population evolving, and is it different from someone down the street with a common cold?
I'm unaware of anybody measuring a quasi-species in the context of a human cold. If that exists, I'd be interested to know about it. But I would sort of pose another question to you, since picornaviruses are your sort of favorite topic. We know, for example, that humans are frequently colonized by these inapparent scaffold-like viruses that have been described in the literature recently. We know very little about that relationship or the consequence of it, only that it exists, and it seems to be a predominantly asymptomatic state. People are looking for disease associates for which you see some claims, but nothing really clear sort of comes out of that. What is the evolutionary pressure and circumstance with respect to that relationship? How do we define that state? If you think about the classical definitions of fitness, then the virus should just sort of replicate as quickly as it can and adapt to something that is allowed to transmit to other populations or other cells. Yet we see there seems to be some inherent mechanism that keeps a lid on this system. How do we explain the evolutionary pressures for that.
I don't know if there's a lid. I don't know if there's enough data to say there is a lid.
The counts of virus that you see in these normal stools, which I think about half of the stools isolated from people have this virus in them, and the numbers are measurable, but they're not enormous. They're kept at some relatively low level. This was my experience when I first got, when I was trained with things like rabies and VSV and these measles, mumps, and negative strand RNA viruses. I was sort of imprinted with the idea: you take a virus, you stick it in an animal, it does nasty things, you get lots of virus,
that's it.
You can propagate it, right?
That's right. That's right.
And then I went and started working with poliovirus, and I thought I'd do the same thing because I saw all these nasty pictures that you'd open up a mouse and it had all these gross tumors from all different kinds of tissue. Polyoma, it's the name of it.
Yeah.
And I thought, okay, I'll purify virus. I made a high titer virus stock. I plaqued it out, and I knew it was good. I stuck it into mouse. I sort of sat back and waited for something to happen, and nothing happened. Mm-hmm. And then I figured out later, well, that's normal. It only happens if you happen to get the right strain of virus and the right strain of mouse within 3 or 4 days of birth, and you put it in here this way, and then maybe you'll get a sarcoma. But normally what happens is nothing. It just goes to the kidney, and it sits there, and you get no apparent disease associated with it. And you and I and most of our audience are probably colonized with the human version of this virus, BK or JC, which we're going to excrete as we get older. And for the most part, nothing will happen. Occasionally, some disease state, like during a kidney transplant or other circumstances, may cause an unusual and rare disease outcome. But, we have 5 billion people on this planet who are probably excreting this virus most every day. And we don't think about that as a significant situation or an evolutionarily important relationship. So that's my area of interest, is trying to understand these much less apparent relationships and what consequences they have.
So do you think in virology these are underrepresented?
Oh, absolutely. Absolutely. If, for example, let's pick a very prominent example, avian flu, which we're very concerned with. There's been a lot of money, a lot of investigation, a lot of thought into trying to prevent the next pandemic. But we've known for 70 years that if you go to a particular population and habitat, certain waterfowl, certain species of ducks, you can isolate the same virus from that population for the last 70 years without almost any variation. Mm-hmm. That tells me that there is a very stable evolutionary pressure for that relationship to be maintained. These are unsick ducks, right, that are they're basically excreting this virus at some low level, kind of like the Saffold virus story in human, mm-hmm. With no consequence for the most part to that duck. But although other birds, you can kill off 200 million other birds that are being farmed as a consequence of a species jump of this virus into that population. What is the evolutionary explanation? What is the HA and what is the NA, these genes that we've studied so much? What are they doing in that context? What is the selective pressure that has maintained them to do whatever it is they do? And there's not a single paper in the literature on that topic.
Well, as you said, people are worried about it jumping into humans, as it does jump into those farmed birds, right?
Yes, and has nasty consequences.
Yes.
Right. But what is the circumstance that's maintaining it?
Right.
Because that's not what you see in its persistent host.
Sure.
What we call a reservoir species. And to me, these are big questions, because these are the relationships that maintain a virus-host relationship for a very long time.
Your point is well taken. Basically, we should be understanding how the virus is doing in its natural host, what pressure keeps it there, and spending less money on worrying when it's going to infect humans. Even
if we understood that, I still think we're probably going to be at a disadvantage trying to predict things. Let's say we were able to go back in history and reconstruct the SARS epidemic emergence. And let's say we had on hand the particular bat, assuming it is a particular species, and it seems to be, and also that might have been a recombinant as well, by the way. That virus is residing in a bat with apparently no consequence to that bat. And if you look at the genetic changes that occurred as it adapted to cause lethal human disease, you see, typically surface protein changes of 20 or so point changes associated with it.
Right.
Had I presented those two viruses and those two circumstances to you ahead of time and say, what does it take to make this virus do this to people? It's a complete mystery. I mean, it's the fact that it happened is clear, but the fact that you could have been able to predict it, I think, is also clear. That there's no science under this for you to actually say, oh, I see. If you do this, we're going to get some kind of hyper storm of cytokines, or we're going to get some vascular disease occur. It's just not a predictable science at this point.
Exactly. Well, I think we could go on with this for quite a while, but we probably should stop. and get you back another time to continue.
Okay.
Yes.
We get a lot of emails at TWiV, Luis, and we like to read them, but we'll just do one today and then give our picks. And here's one from John, who writes, I remember from an earlier broadcast the suggestion by someone that the importance of Ebola, Marburg were overrated by the general populace and that these viruses are overly perceived as contagious. I'm still not convinced. I do have a question, and that is about the physical structure of these viruses. One end being very hook-like has always puzzled me. I mean, I can understand generally spherical structures and even the capsoid injection structure, but why the hook? What does it contribute? Why is it necessary? Why did such an external virus structure preferentially evolve? I hope I expressed that correctly. I understand. Yeah, you've seen these pictures of Ebola.
Yes, absolutely.
Right.
I guess Fred Murphy was the one who snapped the first one, right?
Yeah. I don't know what that means. Do you have any idea?
No, the morphology of a virus is this whole sort of curious topic. It gets to some very fundamental issues of self-assembly that this is an area that is so poorly understood. We just have this view that we understand that if you make similar proteins and they can sort of semi-crystallize and form structures, the reality of anything that's been studied in terms of its assembly is much more complicated than that. So I think trying to guess at the implications as to why Ebola has this sort of curly-Q appearance, I think this is well beyond anything that I can think about right now.
Well, certainly if we could change the particles in some way, we could look at that, but they're so hard to work with, so dangerous, and nobody's doing these.
And remember, like with flu, you have this capacity to assemble it into these morphologically distinct shapes.
Yes.
And that, which has, who knows what, if any, consequences. It's not even clear.
I remember with flu, with fresh clinical isolates tend to have this elongated polymorphic shape, and then when you grow them in cells, then they become more regular and spherical. Yeah, we don't know what that means.
Yeah, it's entirely possible this hook shape is more or less a lab artifact or doesn't have a particular consequence for the virus.
Yeah, I'm sorry we can't do better.
Or it may be crucial to Ebola replication.
Someday someone will find out. A lot of people are starting to use pseudotypes to study all these viruses, which of course is not going to get you any answers because they have nothing to do with the real virus.
One has to be careful with the models you set up to study stuff because you end up being constrained by the models you set up. It's like a catch-22.
Sure.
If you set up a tumor model with polyomavirus, you will learn some things, but that's not going to tell you what T antigen does.
Yeah, absolutely.
And that's what gets forgotten all the time.
Here are our picks of the week. The science blog of the week is Small Things Considered, which is written by Moselio Schechter. You guys have probably seen this one.
Outstanding.
It's writing about microbes to share my appreciation for the width and depth of the microbial activities on this planet. He does a good job. He and one other writer, Mary Jewell, I think is her name, cover a wide range of microbes. So check it out. We have a link in the show notes. Our science podcast of the week is The Skeptic's Guide to the Universe. And this is by a group of people called the New England Skeptical Society. Are you a member of that, Alan?
No, I'm not.
Maybe you should join. Are you a skeptic?
Well, I'm skeptical of a lot of things, sure.
So anyway, they discuss the latest news and topics from the world of the paranormal, fringe science, and controversial claims from a scientific point of view.
That's a topic that interests me as well. People are just prone, scientists included, to have certain beliefs. And the biological basis of that, to me, is fascinating.
So, Luis, if you had one word of advice for budding scientists who are listening, what would that be?
The one skill that science promotes and is dependent on is to be able to objectively understand phenomena and make sense of them. Mm-hmm. And there's a tendency that I think it gets to the same issue, a biological tendency even, to explain things based on rationalizations rather than to interpret the phenomenon that you see. This whole business, for example, with persistence that I've been talking about is a phenomenon that I sort of came across that I just felt needed a lot more explanation. And I just think that's the corrective approach for a long-term career in science. And that's the one that I really encourage people to pursue in the context of their own interest.
Great.
What are the data? What are the observations? And what's curious and doesn't make sense? That should be a point of fascination.
So our science book of the week, I had picked your Viruses and the Evolution of Life, but you think there's a better choice, right? That more recent volume, what is that?
Well, yeah, that's The Origin of Group Identity. But I shall tell you that the last two chapters are going to be sort of an acid trip almost in terms of what the implications of these genetic parasites have to do with human evolution. Because if you look at the activity in our brains of genetic parasites, it's tremendous. And so the last 2 chapters try to offer explanations for that based on how humans themselves work.
Well, we'll leave your book as our pick of the week in honor of your being our guest today, and I'll put a link to the other books so people who are interested in evolution can check them both out. Let's wrap it up. Don't forget, you can find other great science podcasts at 2 other sites, good sites, sciencepodcasters.org and promednetwork.com. Check those out. Luis, thanks for joining us.
It's been my pleasure, and I'm very happy to participate in such an interesting broadcast.
Look, it's about virologists just talking about what they love, so how can it be wrong?
Absolutely.
Come back another time. I got to tell you, though, you have influenced my thinking. Let me tell you what I'm planning to do here. Mm-hmm. I'm trapping wild mice and looking for picornaviruses.
Oh, you're going to find plenty. Tyler's virus, for example, is a really fascinating one from that perspective. It's pretty ubiquitous. It's also one of these ones that if you bring into a colony, it does a number on the colony.
I'd like to find some new ones. And years ago, we talked about this in your office at Irvine, and it's coming to fruition. I've got cages, and now the spring is here. We got mice running, and
well, do you do it in a BSL-3 outfit? I have a picture. I have a student who used to handle wild rodents, and a picture from before Hanta and a picture of after Hanta in terms of how they prepare themselves.
Well, you must know Charlie Kalisher, right?
Yes.
He used to go out into the desert. I've been getting advice from him on how to bait traps for mice and so forth. So
cool. I'm very interested to see what you learn. That's the kind of study that interests me.
I think it needs to be done at many levels, so I'll keep you in touch with what happens. You've been listening to This Week in Virology, the podcasts about all kinds of viruses and where they came from and where they're going. You can subscribe to us on iTunes. Why don't you go do that? And that way you get our new episode every week automatically, or you can go to our website, TWiV.tv. We have a lot of show notes and links to all the things we talk about. And if you have questions or comments, which we always like to get, TWiV@TWiV.tv. Thanks, everyone. Alan, Luis, for joining us.
Always a pleasure.
Me too.
We'll be back next week. Another TWiV is viral.